Today, let us see the chromatographic techniques. Chromatographic techniques are widely used techniques which are used to separate and identify the compounds present in the unknown sample or mixture. Let us see the introduction first. Chromatography is an important biophysical technique that enables the separation, identification and purification of the components of a mixture for qualitative and quantitative analysis. That means it is possible to identify and separate the compounds using chromatographic techniques qualitatively, as well as this technique has been used for quantifying the compounds also. And for the separation of biomolecules like carbohydrates, proteins, fats, we can make use of these various types of chromatographic techniques. Separation is achieved depending upon the various characteristic features of the biomolecules. For example, proteins can be purified based on the characteristics such as size and shape, total charge they possess, and hydrophobic groups present at the surface of the protein, and binding capacity with the stationary phase. And there are 4 separation techniques which are based on the molecular characteristics and interaction type use mechanisms of ion exchange, surface adsorption, partition and size exclusion. Other chromatographic techniques are based upon the stationary bed including the column, depending upon the nature of the stationary bed including the column, thin layer and paper chromatography. And the mode is different from one chromatographic technique to another type. Column chromatography is one of the most common methods of protein purification. Mainly for the purification of protein, we can make use of column chromatography, which is the best suitable method for the separation of proteins. Let us see the principles of the chromatographic techniques. For separating the compounds using this chromatography, we will have to understand the various principles behind the technique, and in this technique we will have to make use of 2 important phases called as stationary phase and mobile phase. Stationary phase is a stable phase and the mobile phase is a moving phase. So chromatography is based on the principle where molecules in a mixture are applied under the surface or into the solid and fluid stationary phase is separating from each other while moving with the aid of a mobile phase. So depending upon the partitioning coefficient of the compounds between the 2 phases, mobile phase and stationary phase, the separation is achieved. The factors effective on this separation process include molecular characteristics related to adsorption, partition and affinity or differences among their molecular weights. Adsorption is adsorption chromatographic type. It uses liquid as stationary phase and solid as a stationary phase and liquid as a mobile phase. And in partition there is a liquid solid phase. And affinity, again you should have both the mobile as well as stationary phase. Because of the differences in using the mobile and stationary phase, some components of the mixture stay longer in the stationary phase and they move slowly in the chromatographic system, while others pass rapidly into the mobile phase and leave the system faster. The following components from the basis of the chromatographic techniques. So what are the important components that form the basis of the technique? First one is stationary phase. This phase is always composed of a solid phase or a layer of liquid adsorbed on the surface of solid support. And mobile phase is always composed of liquid or a gaseous component. So it should be a movable one. And by using this appropriate type of stationary and mobile phase in each type of chromatography, we will be able to separate the molecules successfully. We should know the types of chromatography also. First one is column chromatography, then ion exchange chromatography, gel permeation or molecular sieving type of chromatography, affinity chromatography, paper chromatography, thin layer chromatography, gas chromatography, dyed ligand chromatography, hydrophobic interaction chromatography, pseudo affinity chromatography, and at last high pressure liquid chromatography, and it can also be called as high performance liquid chromatography, which is abbreviated as HPLC. So these are the basic types of chromatographic technique through which it is possible for us to identify and separate the compounds and also to purify the compounds. So according to the nature of separation, according to the nature of compounds to be separated or isolated, you will have to select an appropriate type of chromatographic technique. First, let us see the column chromatography. This technique is used for the purification of biomolecules on a column, that is a stationary phase. Firstly, the sample has to be separated, then wash buffer are applied. You will have to pour the wash buffer in order to carry the sample along the column. Their flow through inside column material placed on a fiberglass support is ensured. So it should freely, the sample along with the mobile phase has to travel down the column without any block. We should ensure that phenomena. The samples are accumulated at the bottom of the device in a time and volume dependent manner. So this is a diagram showing a simple type of column chromatography. Here you can see the separatory funnel at the top, and below that you can see the column, and the compounds to be separated or purified has to be poured into the separatory funnel, and from the separatory funnel it has to be applied onto the column little by little, and separation is achieved inside the column. And when you get the separated components as a zone or layers separately, you will be able to collect it at the bottom by using different vessels or small test tubes. You can collect each and every fraction of the separated compounds from the column after the elution process gets completed. Then, what is the principle behind the ion exchange chromatography? Ion exchange chromatography is based on the electrostatic interaction between the charged protein groups and solid support materials. Here, the stationary phase has to be coated with the ion exchange resins. So, the ion exchange resins are going to be used for the separation actually. Matrix has an ion load opposite to that of the protein to be separated, and the affinity of the protein to the column is achieved with ionic ties. Proteins are separated from the column either by changing pH, concentration of ion salts, or ionic strength of the buffer solution. Positively charged ion exchange matrices are called anion exchange matrices or anion exchange resins and absorb negatively charged proteins, while matrices bound with negatively charged groups are known as cation exchange resins or cation exchange matrices and absorb positively charged proteins. So in this way, the separation of proteins is achieved using this ion exchange type of chromatography. So you will have to take two types of ion exchange resins, anion exchange resins and cation exchange resins. So depending upon the charges possessed by the proteins, they will be attracted either by the cationic exchanges or by the anionic exchanges, through which it is possible for us to achieve the separation. So this is a diagram showing the binding of the molecules to the respective cation exchange resins or anion exchange resins according to the charges possessed by the various proteins. And then comes gel permeation chromatography. It is otherwise named as molecular sieve chromatography. The basic principle behind this technique is make use of dextran containing materials to separate macromolecules based on their differences in molecular sizes. So you may have different molecular sized molecules in a given mixture, and different sized macromolecules will be separated individually by means of using this gel permeation or molecular sieving type of chromatography. This procedure is basically used to determine molecular weights of a protein and to decrease salt concentration of protein solutions. In a gel permeation column, stationary phase consists of inert molecules with the small pores. So the column has to be filled with the gel material which has the different pore sizes. So depending upon the pore size, it will attract the different sized molecules present in the given unknown mixture. The solution containing molecules of different dimension are passed continuously with a constant flow rate through the column. Molecules larger than pores cannot permeate into gel particle and they are retained between the particle within the restricted area. Larger molecules pass through the spaces between the porous particles and move rapidly through the inside the column. Molecules smaller than the pores are diffused into the pores. They will be attracted into the pores, and as molecules get smaller, they leave the column without binding to the pores, and it will leave the column with a proportionately longer retention time. Slowly it will pass on down to into the column. Surfadex G type is most frequently used column material, which is used in this gel permeation chromatographic type. Besides, dextran, agarose, polyacrylamide gels are also used as column materials. And this is a diagram showing gel permeation chromatography, otherwise named as molecular sieve chromatography. So you can see the column filled with the gel beads, and the protein solution is applied at the top of the column, and you can observe the movement of the protein molecules along the column, along the gel beads, and when it settles down at the bottom of the column, you can collect it in various vessels or test tubes like that. Then affinity chromatography. Affinity chromatography technique is used for the purification of enzymes, hormones, antibodies, nucleic acids and specific proteins. The ligand or a chemical substance which has a selective binding with a greater affinity towards the enzymes, hormones, antibodies, nucleic acids and specific proteins are being used in the solid support material or in the column, and they will have affinity towards the substance which is applied as a sample, a ligand which can make a complex with a specific protein. For example, it may be a dextran polymer or polyacrylamide cellulose binds the filling material of the column. The specific protein which makes a complex with the ligand, the protein which is applied which is present in the sample will have an affinity with the ligand which is attached to the solid support. So if the specific protein has the affinity towards the solid support material only, we can get the separation. So according to which the column has to be filled with the suitable ligand which can bind with the specific protein present in the column, present in the mixer with the specific affinity, while free proteins leave the column. So those proteins have the affinity towards that ligand can retain the column, will be retained inside the column itself, and other protein, those who don't have the affinity towards that ligand, will leave the column. Then the bound proteins leave the column by means of changing its ionic strength through alteration of pH or addition of a salt solution. So by changing the ionic strength and changing the pH of the medium, it is possible for us to isolate or separate the bound proteins one by one from the column. And this is a diagram showing this affinity chromatography, how the protein molecules has an affinity towards its binding with the ligand present in the filled in the column. And next comes paper chromatography. This is a very basic type of chromatography. Nowadays it's not being used, but before having a clear understanding on this chromatographic techniques, it is better to perform the paper chromatography in your own laboratory in order to understand the principle of the separation which is achieved using this chromatographic technique, as it is a basic technology. In paper chromatography, support material consists of a layer of cellulose highly saturated with water. In this method, a thick filter paper comprised the support and water drops settled in its pores made up the stationary phase, which is otherwise called as liquid phase. Mobile phase consists of an appropriate fluid placed in a developing tank. Paper chromatography is a liquid-liquid chromatography. That means paper is being used as the stationary phase, and the liquid, the solvent which is used in the paper chromatographic system is used as a mobile phase. Now the mobile phase is going to carry the sample along the stationary paper supporting matrix. Next comes the thin layer chromatography. Thin layer chromatography is a solid liquid adsorption chromatography. In this method, stationary phase is a solid adsorbent coated on the glass plate. As adsorbent material, all solid substances are used in column chromatography. For example, alumina, silica gel, cellulose. They are all used as adsorbents that can be utilized in case of thin layer chromatography. In this method, the mobile phase travels upward through the stationary phase. The solvent travels up the thin plate soaked with the solvent by means of capillary action. During this procedure, it also dries the mixture priorly dropped on the lower parts of the plate with a pipette upwards with a different flow rates. Thus, the separation of analytes is achieved. This upward travelling rate depends on the polarity of the material, solid phase, and of the solvent. So, how to detect the compounds using this thin layer chromatography? If the samples are colorless, fluorescence, radioactivity or a specific chemical substance can be used to produce a visible colored reactive product so as to identify their position on the chromatogram. If the sample is colorless, we will have to make use of some coloring agent in order to visualize the separated compounds. Formation of visible color can be observed under room light or UV light. The position of each molecule in the mixture can be measured by calculating the ratio between the distances travelled by the molecule and the solvent, which is otherwise called as RF value. RF value in all types of chromatography, almost in all types of chromatography, the identification of compounds is done with the help of the RF value. What is called as RF value? Actually, RF value denotes the relative friend. Relative friend means relative mobility and expressed with the symbol RF. RF value is used for the qualitative description of the molecules. So, what are the compounds present in the mixture? For example, if you want to separate the amino acids using paper chromatography or thin layer chromatography, so if you are in order to determine the RF value, you will have to find out the RF value. So, RF value is nothing but can be arrived by the distance moved by the solute divided by the distance moved by the solvent. So, if you measure the distance moved by the solute from the point of origin, from the application point, and you will have to measure the distance travelled by the solvent from bottom to the top in case of the upward movement of the mobile phase. So, if RF value is determined, it is possible for you to detect, identify the amino acids present in the given mixture. So, RF value is the one which will help you to detect the compounds present in the given mixture. So, next comes gas chromatography. In this method, stationary phase is a column which is placed in the device and contains a liquid stationary phase which is absorbed onto the surface of an inert solid. Here, gas is the mobile phase and liquid is the stationary phase. So, through the liquid stationary phase, gas, which is the mobile phase, is going to carry the sample for the separation in the column. Gas chromatography is a gas liquid chromatography. Its carrier phase consists of gases as helium or nitrogen. Mobile phase, which is an inert gas, is passed through a column under high pressure. The sample to be analyzed is vaporized and enters into the gaseous mobile phase. The components contained in the sample are dispersed between mobile phase and stationary phase under solid support. Then next type of chromatography is dye-ligand chromatography. Development of this technique was based on the demonstration of the ability of many enzymes to bind purine nucleotides for Cibacron blue F3GA dye. The planar ring structure with a negatively charged group is analogous to the structure of NAD. This analogy has been evidenced by demonstration of the binding of Cibacron blue F3GA dye to adenine ribose binding sites of NAD. So, it's a it has some limitations. Dye ligand chromatography is not widely used chromatographic technique. Its usage is very limited. The dye used in this ligand binding type of chromatography behaves as an analog of ADP ribose. Just it looks like, or it resembles ADP ribose structure wise. The binding capacity of this type adsorbent is 10 to 20 fold stronger than that of the affinity of other adsorbents. Under appropriate pH conditions, elution with a high ionic strength solution and using ion exchange property of adsorbent, the adsorbed proteins are separated from the column. Then hydrophobic interaction chromatography, which is abbreviated as HIC. In this method, adsorbents prepared as column material for the ligand binding in affinity chromatography are used. Hydrophobic interaction chromatography is based on hydrophobic interaction between the side chains bound to chromatographic matrix. And the pseudo-affinity chromatography, the principle behind this technique is some compounds as anthraquinone dyes and azo dyes can be used as ligands because of their affinity, especially for dehydrogenases, kinases, transferases and reductases. The mostly known type of this kind of chromatography is immobilized metal affinity chromatography, which is abbreviated as IMAC. And last one, high pressure liquid chromatography. You can also call it as high performance liquid chromatography because it's the performance, the efficiency of the liquid chromatographic system to analyze, to identify, to separate, to purify the compound is very high. Its efficiency is very good. So that's why it is named as high performance liquid chromatography. This chromatography technique is used to perform structural and functional analysis and purification of many molecules within a short period of time. So even though expense wise it is superior and it is cost expensive, but the time taken for the separation is very short, and the method of handling is also very simple and efficient in this type of high performance or high pressure liquid chromatography. This technique yields perfect results in the separation, and you can get a very valid or accurate results if you use this technique for the separation. And identification of amino acids, carbohydrates, liquids, nucleic acids, proteins, steroids and other biologically active molecules. Everything is possible with this type of high pressure liquid chromatography. Application of high pressure is the unique feature of this technique. So when you apply high pressure, the sample will pass down the column in a very short period of time to give a very accurate, better results. In HPLC, mobile phases pass through the column under 10 to 400 atmospheric pressure and with a high flow rate. And in HPLC, use of small particle and application of high pressure and the rate of solvent flow increases the separation power, and HPLC and analysis is completed within a short time. That is a unique feature. Essential components of HPLC device are solvent debit, high pressure pump, commercially prepared column, detector and recorder. So these are all the essential components of the system. Duration of separation is controlled with the aid of a computerized system. The material is accurate. And application areas of chromatography in medicine. So what are the applications in the field of medicine? So it has a wider application in various fields, in industrial field, in medical field, in pharmaceutical field, in chemical industry, in agriculture, in research of life science, everything. It finds application in various fields, but here we are focusing the application areas of chromatography in the field of medicine. Chromatography technique is a valuable tool for biochemists. It can be applied easily during studies performed in clinical laboratories, and paper chromatography is used to determine some types of sugar, amino acids in bodily fluid which are associated with hereditary metabolic disorders. In order to diagnose the metabolic disorders, the chromatographic technique can also be used. And in various clinical laboratories for the diagnostic purpose it is being used. And in various research field also related to medicine, the chromatographic technique is a valuable tool you can apply for your investigation. And gas chromatography is specifically used in laboratory to measure the steroids, barbiturates and lipids. Chromatographic technique is also used in separation of vitamins and proteins in food samples. So this is a presentation which covers only the basic principal components of the various types of chromatographic techniques, and in my next presentation we can see the techniques elaborately. Thank you. Thank you for your patient hearing. Thank you.